you do next? Yeah, so now he had these healthy mice and he injected that same agent into the healthy mice. And all those healthy mice start to show the same signs, symptoms, and they all die. So does that prove that it's the same disease yet?
No.
No, because now I have to take a sample from that healthy host that I injected and take a sample from it and be able to isolate and grow the same bacterium that killed the mice. So I know that it's the same bacteria that caused the same disease in my first host and my second host. Yes?
So he does it all over again? He has the mouse and then he takes the sample, then he grows it, whatever. Then he gives it to the other mice and the mice die, then he takes the sample again and he does it all over again?
Yeah, because you gotta prove that it's the same one. I can't just assume that it's the same one because what do we know about pneumococcal disease? What kind of signs and symptoms does it give to humans? Pneumo, meaning what? Respiratory infection. Aren't there a lot of things that cause respiratory infection? You got RSV, you got influenza, you have, streptococci, diplococci, that also cause disease. You have Mycobacterium pneumoniae. All those cause respiratory disease. So I can't assume that just because they had the same signs and symptoms and they died, I have to be able to take that sample out and make sure that it's the same one that was down the same line. So those are his postulates. So you need to be able to explain his postulates. What's the next question? Okay, so you want to be able to explain the work of Semi-Lewis, Lister, and Snow. And they worked in the areas of control and prevention of disease as well as epidemiology. So, who was Semi-Lewis?
He invented hand washing.
Okay, why?
Because he saw that after they took care of the patients that they were getting sick also.
Okay, so he had medical students, and these medical students would go from working with the cadavers to working with real patients, right? And so in between that time, they were not washing their hands. And he, at that time was like, well, people get sick and they didn't know reasons why or whatever. They just thought it was either bad luck or whatever. But the thing is, he noticed that the midwife students, they washed their hands. And they had a much lower rate of infection in their patients than his students did. So he implemented hand washing techniques so that when you go from one patient or cadaver to a patient, you're not transferring that bacteria or any microorganisms to your patient. And if you work in the health and medical field, you probably do this all the time. Every time you walk into a room, you wash your hands. And then you glove up and take your PPE, whatever, and go into the room. When you exit the room, you wash your hands, get rid of the PPE, and move on to where you have to go. And so you break that cycle of transferring infectious agents from one person to the other. So that's what Semi-Lewis did. I'm not going to get on my soapbox about wearing scrubs inside and outside of the hospital. I know a lot of you do. You need to stop that.
Wait, what?
I said, I'm not going to get on my soapbox about wearing your scrubs into and outside of the hospital. I say, I know a lot of you do, and you need to stop that.
Why do you say that?
Well, okay, you're on your way to work, right? And you're in your scrubs, but yet you're messing around with your little kid who might have the sniffles or something, and you get that on your scrubs. You go into that hospital and you're working on the ward, maybe the ICU ward to be more dramatic. And so now you have whatever your 6-year-old just smeared all over your scrubs, but you don't know this, And so you have your hands in your pockets, you're doing things, and you get that in your hands, and you forget to wash your hands or whatever, or even you go into a patient's room after washing their hands, you still have it on you, and say for instance you're leaning over to check something, and you now have contaminated your patient with whatever your 6-year-old has. Vice versa, you work with sick people in the hospital. You leave the hospital with sick people stuff all over you, and you go home, and the first thing you do is pick off your shoes and lay in the couch or your bed. Think about it. That's my soapbox. Anyways. So, Simeon Lewis, hand washing. What did Lister do? Who was Lister actually?
He created the
Listerine, yeah.
He
What's
was
that?
a surgeon.
He was a surgeon.
That treated wounds and his surgery ran with carbonic acid.
Yeah. And so,
Right.
carbonic. Carbonic?
Carbonic.
Carbonic. So he used a solution of carbonic acid, and he would spray the wounds. And he noticed that when he did that, he disinfected the wounds. People didn't end up with, was it scarlet fever? What they were getting? They weren't getting sick after the surgery. So after you've done the surgery and you suture people up, he would disinfect the wound and keep it clean so that he didn't get more infections inside that wound. If people were going into surgery, the surgery was successful, patient died because he got an infection and died. So that's what Lister did. What about Snow? Snow. John Snow. John Snow, he lived in
the well.
The critical.
the well. Okay, what did he find in the well? Vibrio cholera. So how did he find out that vibrio cholera was in his well?
The people drinking water,
Okay, so how do you find this out?
you go to where they're getting the water from.
But how do you know that?
Cause wasn't there like a lady that lived outside of her or something and she got it too and she wasn't like around all those people?
Yeah, so he used the powers of epidemiology. He basically looked at a map of the town and he pinpointed all the people who got sick, and he noticed that they were all in the same area and they all had a central point to where they were getting sick. But then he also had somebody who got sick outside of this area. And so he went and investigated why they got sick, or did they have the same signs and symptoms of the people who were sick in town. And there was. The lady who got sick used to send her daughter down to the well to get that water because she liked the taste of that water better than the water she had. So that one outlier kind of made the questions like, well, why is she sick and she has the same thing these people here have here? So that's where he was able to zero in on the well. Everybody was getting water out of the same well. That's what they had in common. When you're looking at epidemiology, a lot of times you may have a lot of different signs and symptoms or varying degrees of these signs and symptoms, and you're trying to pinpoint one thing that everybody has in common, right? And so that one thing that everybody had in common was they all drink water from that well. And so he shuts down the well, people stop getting sick. They come to find out that the reason why the well was contaminated was because a mother who had a baby who was sick with cholera, she washed the baby's cloth diaper that was with the fecal matter, and that got into the well. That contaminated the well. Now they have vibrio cholera in there. And interesting about cholera is, it is what we call pleomorphic, and it also changes its properties with temperature and nutrient concentration. And so when it's starving and it's not, doesn't have enough nutrients in it, it becomes more and more sticky. And so since there's not really a lot of nutrients in that water, it became more and more sticky, which made it more virulent. And so when people were dumping their buckets in the well, getting the water, and they were drinking this water, then it got into an environment where it could infect people. It was sticking to the cells a whole lot better and causing more illness. But he closed down the well, stopped the problem. He came up with the field of epidemiology, okay? The other people were looking at controlling of and preventing the disease. Okay. In chapter 2, we did talk a little bit about how genes are expressed. So, how are genes expressed?
They're found in DNA, they're related to the protein function. That use transcription and translation to the protein.
Okay. And so remember that central dogma that I wrote up here? Yeah. DNA, RNA. That RNA could be mRNA, tRNA, or rRNA. So, mRNA is your messenger. tRNA is your transfer. And your rRNA is in the ribosome. What's the job of the ribosome?
Like anti-code the proteins and then produce proteins?
It's for the synthesis of protein. So the rRNA is in the ribosome and works with the machinery of the ribosome to synthesize proteins. The tRNA, it carries the amino acid. And that amino acid is specific for the message that is on the mRNA. And so you have a 3-letter code we call codon. So I'll just say AUG, that's the start codon. It's the codon. And then you have an anticodon. That's on the tRNA. And so that would be complementary, so it would be UAC. Right? And so you have DNA, you have transcription. Of that gene, so this contains the gene. So your DNA contains millions of genes, okay? And so if I'm just talking about one gene being expressed, that gene is being expressed through transcription into RNA. And all these RNAs participate in translation. And that translation will produce proteins. So basically, if you take this information here and put it into a essay type form to describe that, would be good.
Okay, so can we say the DNA codes by using transcription and the use of mRNA, RNA, rRNA for translation to the proteins? Pretty much covers it all.
Yeah, but you forgot to mention that was the genes. The genes that
Which is
contain
the
in the
gene
gene.
codes.
Yeah, is going to be carries a code for a protein, and that process starts with transcription of the message from DNA into RNA. That RNA could be a messenger RNA, which carries the messenger, tRNA, which carries the transfer RNA, which carries the amino acid, and the ribosomal RNA, which works in the ribosome for translation. translation occurs and create